Okay, perfect, thanks. So my PhD has really been focused on the human claustrum, and I'm certainly a bit biased by this point, but I really think that this is one of the most fascinating structures in the human brain. So as you can see from this illustration, sorry, the thing is moving in front of me. The claustrum in red is a subcortical and a bilateral gray matter nucleus. And it's most commonly described as an extremely thin sheet wedged between the putamen and the insula, and at least in humans, it's wrapped entirely by the external and the extreme capsules. And in this ventral section that I hope is big enough to see on this large screen, you can see that the claustrum extends into the temporal lobe, where it breaks into these fragmented puddles, or sometimes referred to as fingers. But perhaps the most unusual feature of the claustrum is its remarkable connectivity. It's thought actually to be the most connected structure in the human brain by volume. It has bilateral connectivity with most of the cortex, especially with the prefrontal lobe, and it also has inputs from several subcortical areas. And in this 2005 paper from Francis Crick, they speculated that on the basis of this remarkable connectivity, that the claustrum might actually be a good candidate for a neural correlate of consciousness. And this speculation really kicked off now 2 decades of research in the animal world. I think that it's fair to say that some claims about consciousness have since been tempered a little bit, but it is the case that there's more hypotheses about claustral function, I think, than ever before, including links to salience processing, attention, multisensory integration, sleep, pain perception, cognitive control, and the list really just goes on. But we don't know much about the claustrum in humans still, and its role in cognition. And the main reason is that the conventional MRI that we use most often in research doesn't really afford the resolution to capture that extreme mediolateral thinness that I mentioned. So to illustrate this issue, here's a scan with 1 millimeter isotropic voxels at 3 Tesla field strength from our lab. So as you can see, there's a lot of sort of confusion about where, or ambiguity about where the claustrum might actually be within this region between the putamen and the insula. But nonetheless, a small set of studies have actually measured the claustrum in vivo thus far with MRI, including some at sub-millimeter resolution along the x-axis, and also a few with ultra-high field, which I've indicated with the blue asterisks. And just 12 of these papers report volume estimates that you can see along the y-axis. So although these papers all aim to measure the same structure, the whole claustrum, you can see that the reported volumes are really radically varying. In fact, there's a 5-fold difference between the smallest and the largest estimate. So this clearly exceeds biological variability, and it highlights the difficulty, I think, of reliably capturing the claustrum with MRI. So for my PhD, we really wanted to understand if sub-millimeter resolution MRI at ultra-high field could capture the claustrum in vivo. And to evaluate this systematically, we took a 3-step approach. So the first thing that we did was we mapped the claustrum in BigBrain at the 100 micron resolution. And because this is the first continuous 3D histological mapping of the full geometry of the claustrum, I refer to this as our gold standard. And then we validated this gold standard against 10 Julich brains at the 1 micron in plane resolution. And then third, we measured the claustrum in MRI. And for this, we used 3 7 Tesla datasets. We used a 0.5 millimeter dataset from Montreal from Dr. Boris Bernhard. And we also used a 0.7 and a 1 millimeter dataset, both from our collaborators at Maastricht University. And importantly, I think these datasets are both whole brain datasets that aren't calibrated in any specific way for an acquisition specific to the subcortex or for the claustrum. And we chose this 0.5 because I think it's fair to say that it really pushes the limits of what's available to us at 7T within a feasible scan time. And we chose 0.7 because it's more widely available to a number of centers. It's used by a number of public data sets, including the AHEAD data and the HCP data. And then we used the 1 millimeter, even though we expected partial voluming, like I just showed you on that prior slide. But we used it because it's still the most common resolution for MRI, including for those claustral studies. So I'm going to skip over the details of the methods because of time, but I will mention that for all of these data sets, we did do a manual segmentation of the claustrum. Okay, so here's our gold standard model. Broadly, it's consonant with descriptions of claustral morphology in the anatomical literature. So for example, we can see that the claustrum is a little bit convex because it really hugs the putamen. We can also see that the claustrum is really thin, and even though it's thin, it's not exactly small. So it's actually going about 5 centimeters from front to back and from top to bottom. And its volume is actually quite large. So across both hemispheres, it's about 2,500 cubic millimeters. So this is about twice the volume of the substantia nigra and about 3/4 of the volume of the amygdala, also annotated in the big brain. And now returning to the literature, we observed that this volume is actually a little bit smaller than the most recent high-resolution MRI studies that focus only on the claustrum, which are all indicated above that dashed red line. The red line is the volume of our gold standard. But interestingly, we're larger than all but one prior histological study. And we don't think that this is a shortcoming or suggesting to us that we're actually capturing too much. We think because of the resolution of the big brain and the fact that it's continuous, we're actually capturing the structure as it is. Okay, so we also, of course, confirm, which is a surprise to no one, that the dorsal claustrum is indeed extremely thin. In fact, 85% of the coronal sections contain at least one section where we're maximally 100 microns in width. And we also see that the claustrum is really diffuse. So those ventral puddles at the bottom here are very, very small, and there's quite a bit of space between them. And I think that this is perhaps best captured maybe in this coronal view showing the flat map of claustral thickness. So looking ahead a little bit to MRI, we can really anticipate that these features, namely this thinness in the dorsal section and the discontinuity in the ventral section, will be really difficult for us to resolve. Okay, but before getting to the MRI, we did validate our gold standard in the true histology afforded to us by the Julich dataset. We do observe quite a bit of variability across the 10 brains that we looked at, especially in the ventral section. But quantifying this variability is still a little bit of a work in progress. I have to finish some mappings, so I'm not going to comment on it now. But I think more relevant to the task at hand is that the Julich data actually reveals to us some details that are actually not available in the BigBrain dataset despite the 100 micron resolution. So on the left, we see that the claustrum in red is directly touching both the piriform cortex in yellow and the amygdala in blue. And on the right, I'm showing that claustral cells are also buttressing the olfactory tubercles, shown in purple. And this relationship with the olfactory tubercles, I think, is only mentioned once in prior literature, so it's sort of exciting. But more importantly, we need to know what these neighboring relationships are in super resolution, because it's essentially certain that the partial volume effects will obscure these boundaries in MRI. Okay, so I'll just move to our MRI results, and I'll go through them quite quickly. So here I'm just showing you a representative subject at the 0.5, the 0.7, and also the 1 millimeter resolution. And we're roughly about halfway through the claustrum from front to back. So a surprise to no one as expected as we go to decreasing resolution. The fine anatomical detail that we're familiar with by looking at the gold standard in red is progressively lost. Relatedly, we found that it's only the 0.5 millimeter data set, shown in yellow, that reliably maintains a clear separation from the neighboring structures of the putamen that's shown in pink and the amygdala that's shown in purple. And at the lower resolutions of the 0.7 and the 1 millimeter, partial volume with the adjacent white matter causes the claustrum to sometimes abut those structures, but also more regularly for the claustrum to actually overlap. And it's really a mystery in terms of what structure the given voxel belongs to. So just to look at this a little bit more quantitatively now, we computed a huge omnibus list of comprehensive size and shape metrics, and we compared them to the gold standard. So in these figures, the gold standard is always denoted by that dashed red line. And I know that this is really visual overwhelming, and it's a lot to parse, but the patterns here are all very lawful and very predictable. So the first thing, again, not really a surprise, is that the volume is declining with decreasing resolution in MRI. Interestingly, and something to remember, is that the volume is actually higher in the 0.5 millimeter data set than the gold standard. And we see a whole bunch of variability, meaning that we don't see any reliable measurements of volume in the 1 millimeter data set, which is the most common resolution. We also see that maximal extents in the 3 dimensions are decreasing with resolution. We see that the MRI appears to be a little bit more round with decreasing resolution, but interestingly, the flatness stays constant. And finally, across each two-dimensional slice, with decreasing resolution, we see that both the average total voxel span, so from left to right in the coronal plane, as well as the span of contiguous voxels, decreases with the decreasing resolution. And you might think that this is a copy and paste error because these two figures look so similar if you're paying attention, but it's actually not the case. This is not a mistake, I checked it twice. And you can see that there's a difference in the gold standard measurement as well. So what's happening here is that we're seeing evidence that the MRI at all resolutions we look at is really unable to resolve those fine details of the ventral puddles. Okay, if anybody has any more questions about that one, I'll answer them after the talk. The last thing I wanted to mention is that we looked at the spatial overlap between the gold standard and then the 3 MRI resolutions. And as expected, the volumetric overlap that we see in the boundary precision increases with resolution. And essentially what we find here is that sub-millimeter resolution we think is adequate, but the 1 millimeter probably isn't for most interesting questions that we might want to investigate. And as a sanity check, finally, we downsampled the gold standard to the same resolution as our MRI. And here we see that the overlap metrics in space are much improved. So this indicates to us that it is resolution that's the limiting factor of MRI and not some other fundamental factor like MRI contrast. Okay, so just to summarize in the last seconds, we created this model using BigBrain. We validated it in Julich, and then we investigated the same structure in MRI. And this project left us with the highest resolution histologically informed model of the Claustrum, and we found that sub-millimeter MRI at 7T, we think adequately measures the structure. And we hope that with this resource, and also with these results, we will inspire others, perhaps you, to also look at the Claustrum in the data sets that you might already have. So thanks to my supervisor and our collaborators, many of whom are here. Thank you.